Hello, I'm Ann Montgomery, Editor-in-Chief of BioProcess International Magazine. I'm here at Biotech Week in Boston speaking with several thought leaders about advancements in bioprocessing and cell therapy development. I hope you enjoy their insights and their visions for the future.
My name is Musaddeq Hussain. I work for Merck Research Labs at Kenilworth, New Jersey. It's a R&D labs, and I'm at the biologics part of it, developing bioprocesses for new drugs. And I work at the analytical part. So our facility also produces preclinical quantities of drug substance. So we test in the analytical lab, we test process intermediates and also part of for the release of the drug substance. So that's what I do. So host cell DNA is one of the process impurity because we make the product in living biological cells. So some of the cell ingredients also get co-purified with the drug substance. So we are required to test for the impurities, and the host cell impurities are, say, host cell proteins, host cell DNA, and also the purification column leachates like protein A. So these are the banks of tests that's also included in the release test of the drug substance. For particularly talking about the host cell DNA, it has evolved quite a bit over the last 10 years. It used to be radioisotope-based dot blot estimation or quantification of the residual DNA, but it's sensitive, but there has a lot of issues with using radioisotopes. Then it moved to immuno-based methods, which were not that sensitive. Sometimes we need a very sensitive method. Then we moved into and developed primers and probes for qPCR-based methods. So it's a quantitative PCR. It's very sensitive. Only problem with that is, although the qPCR is very sensitive, it's limited by the sensitivity of the DNA extraction. So if we get a test sample, we have to extract DNA and then put it into PCR. So I think sensitivity and recovery or accuracy of the method are the main issues for all these previous technologies. So in our lab, what we have done is we have changed the paradigm of testing. So the previous paradigm is we get the test sample, we extract DNA, we put it into qPCR, quantitate the impurity. Here is the host residual DNA. The way we change the paradigm, we do not want to extract the DNA. We want to get the sample, put it directly to qPCR by bypassing the DNA extraction part, which was kind of affecting or reducing our accuracy. So we solved the issue of non-extraction related problems, like big molecules tend to coagulate when heated. So what we did, we added protease digestion of the protein part, because we are interested in the DNA. We digest the protein into small molecules. Now they do not coagulate or precipitate when heated. And we did it in a streamlined fashion. We did the digestion in a PCR plate. we denaturation of the protease in a PCR plate, and then we put PCR reagents in the plate and put it to PCR to quantitate. And we published this method, and we have shown that with different drug substance, monoclonal antibody drug substances, the recovery or the accuracy of the method is almost 100%. So as we were developing these methods, we were introduced to digital PCR technology. So this is a new technology, and what it does, if you imagine your PCR reaction happening in a tube, and now take that liquid, say 20 or 30 microliter of reaction, and partition it into, say, 20,000 nanoliter size reactions, droplets. Now you have 20,000 reactions, independent reactions happening. And after, it's an endpoint method, so there is no monitoring or detection of how the reaction progresses. But at the end of the PCR cycling, you look at the droplets. And if the droplets are fluorescing, that means they're positive. If they are not, they're negative. And that's why it's digital, yes or no. And by counting the number of positives and the number of negatives, the quantification can be done without running a standard. So this is a huge advantage for a lab, because the lab doesn't have to carry a critical reagents like DNA standard. And so we started using this new technology in our lab and established methods for PICIA, which we published. And with Bio-Rad's help, we have brought in their CHO residual DNA kit and established in our lab. So one improvement we have done over that method is, like the qPCR method, we introduced a protease digestion in front of it, and that allows us testing about 100 to 150 microgram of drug. The original Bio-Rad kit allowed far less than that, but if we can test more drug, it's advantageous to us. Well, for example, the direct method, the method I described, where we directly put the test sample into qPCR without DNA extraction, but by incorporating a protease for digesting the drug. That's a new technology that every lab that does host residual DNA from biologics can use that technique. And that will reduce their work time, importantly, increase their accuracy. So, and also, digital PCR is a new technique. It's not very difficult to do. There are, for example, the droplet digital PCR. Bio-Rad has automated instrument to make droplet. So you mix everything and feed it to this droplet generator. You get the droplet, you run the PCR, you get a results without running any standards. So these are the techniques that labs, especially new labs, can incorporate and benefit.